Hello everyone, welcome to the Bodhi Academy. In today's video, we are going to start with the new chapter from 12th Biology, that is chapter 11, Biotechnology, Principles and Processes. So let us begin. First of all, what do you mean by biotechnology? So it is a field of biology that deals with the use of live organisms or the biological systems to make products or processes that will be useful to human beings. So it is a field of biology that deals with the use of live organisms or biological systems to make products or processes that are useful for human beings. Now let us see the principles of biotechnology. So there are two principles, that is genetic engineering and bioprocess engineering. What happens in genetic engineering is that it is a technique to modify the genetic material. It is a technique to modify the genetic material, means the genes of an organism. It allows the introduction of these modified genes. So the genes which are modified, it allows the introduction of these genes into host organisms. Now after the introduction of these modified genes into the host organisms, it will change the phenotype of the organism. That is, it will change the phenotype of the host organism. So there will be some changes that will be seen in the organism. Then talking about the bioprocess engineering, it is the maintenance of sterile conditions to enable the growth of only desired microbes. Sterile means a condition which is free from microbial contamination. So it will not allow all the microbes to grow. It will eliminate or eradicate all the microbes which are present in that area, and then it will allow only the growth of certain desired microbes. So it prevents the growth of undesirable microbes. Then talking about some of the basic concept of biotechnology. So now we have talked about genetic engineering as modified genetic material. Okay, technique to modify genetic material and then introduction of that genetic material into the host organisms. So what happens when a DNA molecule is somehow introduced into a host cell? So what will happen? Now simply introducing the modified genetic material into host cell, it will not replicate. Okay, so DNA cannot replicate within the host cell. It will not be able to self replicate, and therefore most probably it will be degraded. So how can you make the foreign DNA multiply or make copies of itself within the host cell? So this is our next question. Now what we need to do is, the foreign DNA must become a part of the host organism's genome. Genome means all the genetic material of the host organism. It has to integrate in the gene or the DNA of the host organism. Once the foreign DNA is integrated into the host organism's genome, it now gets multiplied along with the host chromosome, as the host chromosome will contain the origin of replication. Now again the next question is, what is origin of replication? So origin of replication is a part on the gene or the DNA which is essential to initiate the process of DNA replication. Basically, origin of replication is the start point which is essential for process of DNA replication. Then this process is also called as cloning, or making multiple identical copies of any DNA templates. Then let us talk about the construction and cloning of the first artificial rDNA molecule. That is, rDNA means the recombinant DNA molecule. Now let us see the steps involved. These were the scientists that constructed and cloned the first artificial recombinant DNA molecule. So let us see the steps involved. The identification and cutting of piece of DNA from a plasmid that provides the resistance to antibiotics. So the desired quality or the trait that we have taken is resistance to antibiotic, and this we need to introduce in a host organism. So first what we'll do, we'll identify and cut the piece of DNA from the plasmid of bacteria. So how do we cut this piece of DNA? So the DNA was cut using cut at a specific position using the enzymes, okay, and those enzymes which are used to cut a DNA or a piece of DNA are called as restriction enzymes. They are also called as molecular scissors. Now we have talked about a plasmid. So what exactly is a plasmid? So a plasmid is a double-stranded autonomously replicating, that is self-replicating extra chromosomal circular DNA. So it is basically like this. In the bacteria, a circular DNA that provides the additional features to the bacterial cells if present. So if these plasmids are present in the bacterial cell, they will provide additional features to the bacteria such as resistance to antibiotic. Now here what we have done is, we have cut a small piece of this plasmid using the restriction enzyme. So this is a piece of DNA that we have cut. Then let us see the next step. Now the cut piece of DNA was linked to the native plasmid of this bacteria, E. coli, and the enzymes involved were DNA ligase. Now those enzymes which are used to join or link the DNA molecules together are called as DNA ligase or DNA ligases. So now we have to introduce this cut piece into the host native plasmid. So suppose this is the host DNA. We have to introduce the cut piece which we have cut over here, and then we have to join these two. So let us see this modified circle. Now after using DNA ligase, these two DNA molecules will be joined together. Now this modified circular DNA is a recombinant DNA. Now since we have combined two parts of DNA together, it is called as rDNA or the recombinant DNA. So, and it acts as a vector. So first of all, rDNA, a recombinant DNA, is a DNA molecule that has been modified and may have a foreign DNA segment. So over here, this part is the foreign DNA segment. Then what is a vector? So vector is a carrier of DNA or foreign DNA or gene of interest. So this part which we have introduced in the host genome is our gene of interest, that is antibiotic resistance. Then talking about the next step, now upon introduction of this rDNA into the Escherichia coli, that is E. coli bacteria, the rDNA was able to replicate within the host cell and multiply in number using new host DNA polymerase enzyme. So then again after this, the DNA will be multiplied. So as the original antibiotic resistant gene has been multiplied within the E. coli host, this process is referred to as the cloning of antibiotic resistance gene. Now what is the antibiotic resistance gene? It is the gene that enables the bacteria to survive in a medium containing antibiotics. Now let us see the three basic steps in genetically modifying a organism. So the first step is identification of DNA with desirable genes. So first step is identification. Then the next step is introduction of this identified DNA into the host organism or the host cell. Then the third is maintenance of the introduced DNA in the host and then transfer of that DNA to its progeny. So we have to see to it that the introduced DNA molecule is then further transferred to its progeny. So this is all in today's video. Hope you have understood this. Thank you for watching my video. Please like, share and subscribe to the Bodhi Academy.